Okay, so I'm going to just make a couple of preamble remarks here as we allow people to gather. This is the beginning of the hour. We're starting on time. Please, I suggest that you select the speaker view in the top right corner, at least on the Windows Zoom. You click on view and select speaker view. That way, whoever's speaking will be who you see. That makes it tend to work a bit better. We will be recording this webinar entirely, well, hopefully entirely, and we'll be producing a couple of versions of that that you can refer to afterwards. One which will have a clickable index on it, so you can re-listen to the parts that you care about most. So we'll be doing that. And okay, so I'll get started here. My name is Rod Turner. I am the president and founder of Manhattan Street Capital. We are hosting this webinar. I'm not allowed actually by regulations from the SEC to pitch any particular company, so I won't be doing that. But what I am doing is hosting the event and helping and interviewing to some extent and asking questions of Greg and Tommy during this session. So I'm going to introduce Greg and Tommy right now. Please read the disclaimers that are in the chat. And also, please post questions that you have at any time in the chat section. At the end of the webinar, we will be answering the questions in order, as well as bearing in mind where we can add the most value by answering those questions. So please feel free to start putting them up as you see fit. So I'm pleased to introduce Greg Hummer, who is the CEO and founder of IdentifySensors Biologics, and Tommy Soares, who is the Assistant Director at Purdue University of Infectious Diseases, and a lot more that I don't want to spell it all out because it's such an incredibly lengthy and impressive title. He's a doctor, as is Greg. They're both doctors. I'll leave it at that. So I'd like to introduce them. Greg is going to present the company and Tommy will as well. And then we'll get into answering questions from the audience. We expect the entire webinar to last for approximately 1 hour. Thank you very much. Welcome here. Welcome joining us. Look forward to hearing from you. Hope you find this to be a very useful event. Over to you, Greg.
Well, thank you for joining us all today. We're here because of a very important issue regarding a pandemic. We know that there's a lot of information going out today through the television and radio, and a bit of it is confusing. But we're here today to dispel any confusion amongst testing. We believe that our Check for COVID test is a very unique test, because it is a self-test, a truly self-test done within your home or on the go. And it's very inexpensive so that it can be done multiple times a day or multiple days a week, if so needed for your business or your school or your travel. So we're here today to answer some questions from everybody. We will describe the test, where we are today with it. Dr. Soares is here. By the way, I'm a medical doctor, and Tommy Soares is a PhD. So Tommy is here today to talk about the technical aspects of the test, and I will handle all the other aspects of the test related to marketing, production, and how it works. So Tommy, want to introduce yourself there?
Sure. Thank you, Greg, and thank you, Rod, for hosting us today. My name is Tommy Soares. The whole mouthful is I am the Assistant Director of the Purdue Institute of Inflammation, Immunology, and Infectious Disease at Purdue University, where we are developing these sensors, specifically the Check for COVID sensor today. And we've created this partnership with IdentifySensors Biologic to bring this technology to you, and we hope to be doing this in an accelerated pace, knowing that we're all dealing with this pandemic, and this can be a great solution. It's probably going to be a real solution for this pandemic. So thank you so much for having me.
Thank you, Tommy. One of the things that we get a lot of questions about is how is this test really different from all the other so-called at-home tests? And it's markedly different, it's radically different, and here's how. The current at-home tests really aren't done at home. Current at-home tests are usually collect saliva in a tube, send that tube in to have an RT-PCR test, which is the gold standard. Our test is unique because it's simply done by yourself on your unprepared saliva at your home or on the go, for that matter. And it's done in a reader in the size of the palm of your hand. And it's very inexpensive. So it can be done at home by yourself. And it's an electronic test. It's not a chemical test. And Tommy will go in and explain to you the difference between an electronic test and a chemical test. Tommy?
Yeah, so thank you, Greg. One important feature about this test is the speed at which we can detect the virus. And the difference is that, as Greg mentioned, the saliva that you collect at home and you send to a lab, well, when you collect that saliva, it's chock full of preservatives in that kit that you are sending to the lab. And so they have to deal with those preservatives. They have to deal with that saliva in steps to extract the genome of the virus and then conduct this RT-PCR test, which involves chemicals, it involves enzymes and reagents, and that's why it's so costly and so time consuming. Our test is based on nanotechnology techniques. And this nanotechnology, what I really mean, it's a particular science that allows us to really peer down beyond the microscopic level, down into the deep molecular level, and start seeing these molecules, detecting these viruses. And so our results would be very quickly, within minutes, and you're going to get those results displayed on a smartphone device back to you. So you, as Dr. Hummer mentioned, these tests can be done really anywhere, at home, at school, at the airport, wherever you might be, wherever the test is available. If you have a smart device, you should be able to and obviously the reader with the sensor, you should be able to see if you're infected or not. And the difference is that because we're not using any preservatives, we're not using any extra chemicals, we're able to reduce the price point, we're able to increase the speed, and be able to deliver a product that is actually going to be more helpful than what's out there. If you think about taking days to get your results back from these typical labs, rather than being able to see that result within minutes on your smartphone, well, we'll be able to more accurately detect who's infected, where these blasts of infections are occurring, where the hot spots are, and how we can mitigate those transmissions quicker and more accurately.
Thanks, Tommy. The second most prevalent question we get is, well, I've heard about these antigen tests and I've heard about these paper tests. What are those and how are you different from those? A paper test is like a quick pregnancy test. It's a chemical test, still relying on chemicals and other reagents. However, in that test is testing for surface antigens of the capsule of the virus, okay? And those surface antigens have commonality to other coronaviruses that are relatively harmless that cause the common cold. So they're more prone to give you a false positive test. And you've known about false negative tests. Those are the ones we don't want, false negatives. Of course, in today's world, a false negative can be as bad as a false positive. And here's what I mean. Let's say I'm the governor of Ohio and I want to meet with the president. I come up to Cleveland and I have a rapid paper test. and it says I'm positive. Well, this is a true story. So I'm Mike DeWine and I can't meet with the president because I had a fast test, antigen test. He later goes back to Ohio State Medical School and has an RT-PCR, which is negative. So having a false positive test may be, in today's crazy world, as bad as having a false negative, meaning you have to cancel your plans, you have to quarantine yourself, and so on and so forth. So we want to have a test that is accurate, that is dead to rights accurate. No false positives, no false negatives. We don't know if that test exists, but we're as close as you can to that because of the way the test is constructed and the fact that it's not a chemical test. It's a molecular test. You've heard Tommy refer to a molecular test. Well, a molecular test refers to detecting the genes. And there are two genes that are extremely specific for COVID-19 virus, and that's gene N1 and N2. So we have functionalized our sensor to detect those two genes. When those genes are there, we get a positive hit, you get an accurate reading. If they're not there, so far, we have no false negatives. We're going to be going to using clinical studies, actual clinical samples very soon in the BSL-3 lab. That's a biosafety level 3 lab at Purdue. Purdue has all of the necessary tools and facilities that we need to do this properly. So we are a molecular test. We test for the genes. We are not an antigen surface test, which is very important. The other thing with antigen tests is they have to be interpreted by the human eye. It's usually a colorimetric test. So the FDA really isn't excited about a test that maybe 300 million Americans have to interpret themselves. Our test has a digital output. And because it's a digital output, we can feed it to a cell phone app. The app then feeds it to the cloud, where we operate and look at the data in a mathematical formula, and we decide if that test is positive or negative. And then we can do automatic reporting back to you or to any smartphone, and also to the CDC and the appropriate state health agency. Tommy, at this point, maybe you want to go into a little bit more how the test is differentiated from a surface antigen.
Yeah, and thank you. I think you described it well, Greg. And I just wanted to indicate that, you know, one of the biggest troubles that we're seeing the FDA is having is just the sheer number of tests that have come out and that they've had to approve on the RT-PCR side, and now all these antigen tests that are coming out. You know, the antigens tests are detecting, as Greg said, the outer shell of the virus. These are proteins, like you might have heard of the spike protein, the coronavirus, the crown, or these spike proteins that jet out of the capsule of the virus. And it's those particular spike proteins that are being recognized in these antigen tests. And because there are many different types of coronaviruses, like you might have heard of SARS or MERS, or even just the regular coronaviruses that give our kids, you know, the sniffles at school, those all have this spike protein. And so there is a higher chance that these antigen tests might also cross-react to the coronaviruses that are of common cold. What we're doing, as Greg said, we're peering deep into the genes of the virus, and we're detecting those particular ones. And we're detecting them very accurately. We should be able to tell you the difference if you have a SARS infection or if you have a SARS-CoV-2 infection or with other viruses. And I know there's, you know, questions about the different variants that are circulating, this secondary variant that we're seeing in Europe. I think our tests, because of the way that we're designing these sensors, we are going to, I'm fairly certain that we're going to be able to at least get to that intended gold standard of being able to differentiate between the different variants. And that's obviously the ultimate goal here, is not only be able to tell you if you're infected, but be able to tell you if you're infected at an early stage of infection and tell you what is that infection that you have.
I think Tommy hit on the most important point, and that is, don't we want to know early on that we are carrying a virus that can be transmitted to other people? We all know that in an early viral infection, you have very few symptoms, if none at all.
That's
So,
right.
and it's so too with this virus. In fact, you've probably heard on the news that there are people who believe that because this virus is a unknown virus to most of humanity. So what we're looking at is people who have differential symptoms to the virus. I might not have any symptoms for 2 days, but I'm still spreading it. Whereas someone may come down in just 1 day with horrendous symptoms. So you want to be able to tell on the front side, do I have the virus? And if I do, even though I don't have symptoms, I should stay home from work, I should stay home from school. This leads us into the next big question that we get, which is the vaccine for this virus going to mitigate against your product being successful? The answer is absolutely no. The vaccine, as we know it, was supposed to already inoculated 20 million people. It's only inoculated 2 million people. There's going to be a lot of people that don't take it. And then there's going to be the problem that, and this is according to the FDA, when they approved these vaccines, they did not know how long they would be useful for. So what we're finding out is in actual infected people who have the disease, their immunity wanes in about anywhere between 5 and 8 months, which means they have to get another shot, which means you can get this twice. And there's already been cases of people getting this twice. So this is not to scare anybody, but this is the fact of this virus. So as an adjunct to a vaccine, we believe that an accurate self-administered test at home, a truly at-home test, a molecular test, can tell you if you can go to work or not, or go to school or travel, or at least stay away from people if you're positive. And a lot of the antigen tests will not be able to detect early virus loads like our test will be, because we're a molecular test. So the bottom line is, and Tommy, you may have some ideas on this yourself. The bottom line is we're going to need a test like this, and we're going to need it for at least the next 20 months. I don't think this virus is going to go away very quickly. You can tell that we're not being vaccinated at the rate that we should have, and a lot of people aren't going to take this vaccine, and that's just going to be the fact of it. So Tommy, do you have any thoughts on that?
Yeah, Greg, I think it's a very important point because, you know, I don't know about you, but now since COVID has come around, whenever I start to feel a scratchy throat, or I start feeling like, oh my gosh, I'm getting a little sick, I might have a cold or something like that, I don't automatically think I have a cold. I'm probably like most humans, I start to fear that, oh my gosh, I have something else. And I'd like to know immediately what that's something else, to make sure that it's not SARS-CoV-2, that it's not COVID.
Right.
And so this test will give you that peace of mind to know that you or your family member who's just come down with a scratchy throat does not have SARS-CoV-2 infection, or does not have that COVID-19 infection. The other thing, Greg, is that most tests out there you have to go out and go to the clinic or go to the testing site to get a nose swab, to get a, something like that going. Or as we said, you might collect your saliva and then send it out into the lab, and you're waiting and waiting and waiting. And if you do get that result back after 3 days and you're fully infectious and you've moved around, you've spread that around, and you've also probably put yourself in danger if you haven't sought medical help. The availability of having this result report back to you on your smartphone now will allow you then to access telehealth networks and be able to access medical attention if you need it at the palm of your hand. So with a positive result, you'll be able to mitigate a little bit better how you deal with that situation with our tests. And I'm fairly confident that that is going to bring a big component to this, because if I need to speak to a doctor and ask them questions because I don't know what to do next, if I have a positive result, well, I can access that information and speak to that person immediately.
Tommy, that brought up a good point, and that point is that early treatment is also an incredible valuable aspect of this test. It's not just if you're positive. I believe as a medical doctor, getting appropriate early medical treatment will keep people away from the hospital and certainly keep people out of the ICU. And having a test where they shove something up your nose and you get the results back in 5 days. Remember, by the time you have symptoms, you probably already had this virus for 3 days, maybe even 5. So you're 5 days into it, then you wait another 3 to 5 days to get the result. By then you're sick. And then you're questioning, do I have COVID? And then the fear factor comes in. You rush to the hospital, you rush to the emergency room, and the propensity, the bar is very low for admitting people into the hospital today with COVID for various reasons. So early treatment is key. Now, since we're entering the flu season now, 9 out of 10 times when a person gets sick in the United States, they're probably not going to have COVID. They're going to have the flu, but they're going to think, like Tommy said, oh boy, I've got COVID. What am I going to do? And if anybody's listening here that's had the flu, the regular flu, it's not nice. You're very sick. In fact, we had 43 million people last year in this country actually die of the flu. So this brings up another very important question that people ask us all the time. In fact, there was a company that we spoke with recently that said, can you put more than one test on a cartridge? Now we have the reader and the cartridge goes in the reader. So right now we're working on a single test on the cartridge, which would be for COVID-19. However, we've planned this in such a way that we can put up to 6 different pathogens on one cartridge. And what the first one that we intend to multiplex, that's the term given to being able to test for multiple diseases or pathogens at the same time. It's multiplexing. Given the fact that we can multiplex up to 6, the very first one that we want to multiplex the virus is influenza A and influenza B. So in the near future, you'll not only be able to tell if you don't have COVID or you do have it, but you'll be able to tell if you have influenza A or B. So this is a platform technology. It's not just a one-shot test. This is a platform technology that we will develop to diagnose rapidly at home in your own house for different viruses and bacteria. Those viruses will be influenza A, influenza B, Zika, dengue fever, and then other viruses such as hepatitis C in the future, and even Lyme disease. So I don't want you to look at this as just a one-shot deal test like everything else in the market. This is a very unique opportunity. It's a select opportunity instead, in fact, because this allows us to put a laboratory in your medicine cabinet. And that's the way healthcare is going to go in the future. And I won't say that we're going to disrupt healthcare, but certainly healthcare and laboratory medicine has been stuck in the 50s and 60s, and we're about ready to change that.
Well,
So,
Greg, I just want to add in,
sure.
since you were mentioning all of these other infectious pathogens, influenza A, influenza B, I don't know about you, but I've gotten certain questions regarding how do you have access? How can you work with all these dangerous pathogens? And as you were talking, I was really thinking a little bit about the history of how we got to make these sensors at Purdue. So I work very closely with the director of our institute, Richard Kuhn. He's a virologist. He studies viruses of all different kinds. You might have heard his name before because he was the one that deciphered the structure of Zika virus when it first came out, and he published that in a fairly widely circulated journal. Um, and since that beginning of knowing the structure of Zika and working with Zika, he's actually, his claim to fame is being the first one to decipher the structure of this awful virus, also called dengue. Which is more found in tropical areas. And these are viruses that are transmitted by mosquitoes mainly, dengue, Zika, you might have heard of. And that's his strength. He really works and knows these viruses well, and he has collections of them in what are called biosafety level 2 and biosafety level 3 laboratories that we have at Purdue. These are very specialized laboratories that allow us to work with these dangerous viruses. And in his group is actually, these are the world experts in being able to manipulate these viruses and grow them and ask questions, biological questions about them. And for many years, since actually 2015, he's been working in close collaboration with Dr. Leah Stanshu, who's a professor in materials engineering at Purdue. And so here we're bringing biology with engineering experts, and we're fusing these together to create these new types of sensors, this new type of technology. So the technology really was started to be developed for dengue, for Zika detection. And obviously, because of the opportunities now to bring solutions for this COVID pandemic, we're really focusing deep now in detection of SARS-CoV-2. And so the resources that we have both for the biosafety level 2 and biosafety level 3 laboratories, the biology, all of those resources, microscopy, and everything that we're going to need there and that we are using now also allows us to bring some of these other dangerous pathogens like influenza, like hepatitis C, which Dr. Kuhn is also working on. And on the other side, we have the engineering, huge engineering strength at Purdue, where Dr. Stanshu is really resourced by the top labs for nanotechnology, microfabrication, and roll-to-roll scale-up capability. So, one of the questions people have is like, well, how can Purdue play a role in manufacturing? Well, what we can do is bring out the resources together and start creating the prototypes and start developing them in these platforms. For example, the roll-to-roll capabilities that we have that will allow us to test now a scale-up production of these sensors. And all of these resources are all in one place. The Burke Nanotechnology Center at Purdue's Discovery Park, the Binley Bioscience Center, and Richard's laboratory, Dr. Kuhn's laboratory, with the structural virology area. So we're putting all of these, synthesizing them together to bring forward some solutions to this real complex problem.
Greg, could you talk to the schedule, the process? That one of the questions, we've got a few good questions actually waiting, but that one would be perhaps a good one to address now.
Greg? I think Greg is frozen. I can just jump in here, Rod, if everybody can still hear me. Um,
Yours
eh,
is, you're working fine.
okay. Yeah, I see Greg dropped off. I don't know if you prefer us to wait for him to
Oh,
join
no,
back
keep
in.
going. Keep going. He'll be back in a moment.
Right. So we are aiming to be in front of the FDA within 60 days. It's a lofty goal, but there's so many people working really hard to make this happen, and we've made really great progress in the lab so far. That right now we're really trying to optimize that piece for handing off to manufacturers. So the FDA within 60 days is a goal we're striving for, and that would put us in the manufacturing probably towards the end of March.
Can you hear me now?
Yes.
Yes. Hi,
Okay.
Greg.
Where are you?
I lost my feed.
Yeah.
Yes, and
We can't see you, but we can hear you.
Okay. Well, maybe, I don't know what that is.
it looks like you're
Um,
pointing the camera the other way.
Yeah, the camera's faced, there's something up with the camera. Just you could switch back to the original camera, or I don't know actually what to do, Greg.
Yeah, it's okay, but we can hear you, Greg. Probably more important, you're a good looking, there you
There you
go. Nice.
go. You're back. Well done.
Perfect. I was gonna say, we're a good looking guy. You're a good looking guy. We know what you look like, but go
I did
ahead.
want to say something about Richard Kuhn for everybody's edification here. Richard is a world famous virologist.
So lean into the computer because your voice is a bit quiet, Greg. Thank you.
Yeah, Richard Kuhn is a world famous virologist who actually is the department head at Purdue of virology. And Richard is fully behind this program, although he has no monetary involvement in this company. He is an advisor to the company on the advisory board. So I just wanted that to be plain and clear. But he is a super guy in this area of virology. So, Tommy, that was a good explanation of multiplexing and the value of the platform. I did want to say one other thing about the multiplexing issue, and that is that the reason we first got started at Purdue was we were looking for a test for fruits and vegetables. See, we have another vertical in the company that actually is going to be putting these sensors in fruits and vegetables so that we can prevent E. coli outbreaks like in lettuce and so forth. And it turned out that this test was an ideal test for that. So right now we're doing it for COVID-19 and other viruses, but in the very near future, we'll be doing it for other bacteria like MRSA. MRSA is a $20 billion a year problem in hospitals. That's methicillin-resistant Staph aureus that has plagues hospitals throughout the world. And we'll be making it for E. coli, for lettuce and fruits and vegetables to be tested. So that's how we really got connected with Purdue, because Purdue is a big agri hub for technology. So the chairman of the board, Mike Berghoff, called me one day and said, well, we have this super group of people that's at Purdue doing this particular test. Could you come and help us out? And we had already done most of the electronics work for the test. So we paired up with Purdue, and Purdue is producing the sensors and we're producing all the electronics. And so it was a great marriage and it's working out really well. And we're very happy with Purdue. And we're going to continue to work with Purdue and build a mini like Manhattan project of very educated PhDs to continue to formulate these sensors for all the different types of things that we want, be it Lyme disease or MRSA or E. coli in the field on lettuce and things like that.
So, Greg, one of the questions that has been asked is about the specificity and sensitivity of the test. Can you talk to that?
That's a great question. I'm going to let Tommy handle that one.
Sure. You know, The specificity and sensitivity are all related to how much can you detect, and how specific or how much of that particular variant or that particular pathogen you can detect. Well, our particular test doesn't require any other chemicals. We're using these nanotechnology techniques to really measure minute quantities, and we are we are probably better, or at best, as that gold standard is, being able to detect, several hundred copies of the virus. So, and that's what the RT-PCR test, that is the gold standard, that's what they are measuring. And there are even some out there that are not as good as some of the ones that are performing better. But we are because of this nanotechnology techniques that we're using, we're able to identify minute amounts, and it's very specific to SARS-CoV-2. The best part is that you don't really require any clinician or any healthcare worker to administer the test. You don't need to go anywhere. Essentially, you can just be where you are and test yourself anywhere. So really, this is a test for anyone, anywhere where they are. And the specificity and accuracy that we're reaching is that gold standard.
Yeah.
Could you shed light on the process of getting approval from the FDA for a test of this type?
Sure. So there are different, and we're gonna be presenting to the FDA a bit of a different technology. So we'll see how they react to us. But in essence, it's not going to be that much different than what they've already started to approve. And so the process is, you have to show a certain level of accuracy with so many tests that come back. And the cool thing is, as Greg said, the cartridges, those are going to be one-off, but the reader is reusable. So you should be able to take, 20 cartridges from 20 infected people with one reader and be able to detect each one of those, or with 20 different readers. So what we need to demonstrate to the FDA is that we are fast, we are as accurate and as specific and as sensitive as that gold standard. Once we're able to show that, and I'm fairly confident that we are actually going to be reaching uh, that goal, that I think they will let us pass through. Because if you think about it, Rod, the 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 advantage of check for COVID is that the time is diminished. The cost is diminished. If you think about what it costs to take a saliva sample from your home, put it into a preserved cartridge, mail that off to a laboratory where a specialized scientist is going to take that sample, is going to treat it with chemicals, it's going to put it through a process with more chemicals, more detection, and then they have to upload that information to report it out to the CDC and so on and so forth. We're going to be able to completely uh, diminish the amount of time that all of that would be done without the need for a clinician, without the need, any person will be able to do this. And so I really think that the FDA is going to look at that and is going to say, you don't need anybody to interpret the result. And so if you don't need, like, this is the problem why we don't have any home tests, is because they don't, as Greg said, they don't want to let 300 million people interpret the results. Is this really positive? Does it look positive to you? Was there a color change, or does it look like it is or not? And so we're going to take all of that away. They will not have to do any interpretation. And so that should accelerate and smooth and pave the way for FDA approval for this type of test. The speed the speed at which we're going to be able to deliver results is going to be monumental, because now in real time, we will know who is infected. And the models for transmission that the CDC has been trying to use, these epidemiological models that people use to look at the rates of infection, how many more are we going to get, where's that curve, and so on. All of those models are absolutely flawed, because we have to wait 3 or 4 days or even 10 days to get our results back. And in the meantime, we've touched people, we've moved around, life has happened, we've continued living, and the transmission rates are through the roof. And so, unless you have a test like Check for COVID that can report in real time what the status of infection is, we're really not going to be doing, and as we can see, we haven't been doing much of an effect on this pandemic.
We've
Please
been,
shed light on there's a couple of questions here broadly on the same topic, which is, will this test be possible to use by the airlines in schools and for traveling, work, etc., things of that type?
yeah, can you hear me?
Yeah, you're a
Yes.
little faint, but we can hear you.
Just wanted to add one thing there. We've been in touch with the FDA all along. They've given us a special template. They're looking forward to our data, and we should have that data sometime within the next 60 days. And I don't think we're going to have any problem. The FDA is literally approving a test that probably should not be out there, but they have no choice at this point. So
Yeah.
the answer to your question, Rod, is yes. This will be a huge, huge advantage for travel companies, airlines, cruise ships, schools, employers, and just your everyday family, because they'll be able to now control their destiny and do this test, whenever they need to. Like I said, our importance is on the front end of the disease, telling you when you have it as early as you can. And because we know what happens over a 14-day period, you generally will clear the virus. Some people, though, don't clear the virus for as much as 20 days or more. But yes, we will. That'll be a big help to those market sectors.
I will add also, Greg, I know we're focusing on Check for COVID test here, but I know you also mentioned the food pathogens vertical, E. coli, Listeria. One of the big advantages is not only will we be able to protect the food supply, but also the people that work in that food supply industry will be able to protect those employees as well. And so again, having that type of perspective for this type of technology, we really hope that and we know already that it's going to bring a major solution for all of these problems that have been almost unintended consequences of this pandemic that have risen up in this past year, and we've all started to feel it.
So one of the questions also is relates to schedule, which is, once okayed by the FDA, how quickly can the company ramp production?
Yeah, that's a great question. We've taken a lot of pains to make sure that the components of our test are readily available, number one, that they have shelf life, long shelf life, and that we can get to the market quick and manufacture these quickly. We're happy to say that all the components but one are manufactured within the United States. The chip is an off-the-shelf chip produced by the hundreds of millions in the United States. There's one component having to do with the cell phone connection that will be purchased from Norway. We've identified several manufacturers that we can quickly spool up. We're also hoping to get a BARDA grant or a national health and NIH grant from RADx. These grants are particularly specific for commercialization. So we've got some really great engineers at Purdue and engineers on our staff that are very confident that we're going to be able to spool this up at the end of March. That's our shooting for date.
So there's another question which is specific on schedule, which is how soon will the prototype be finished at the laboratory at Purdue?
Question. We have our electronics guru wizard, Rod Corder, working on that as we speak. We are due to get a full prototype by the end of January, maybe first week or two in February. Keep in mind that in the lab, we have successfully produced the electrodes that will go in the cartridges, and we know how they work. We're just trying to functionalize them to be even better at this point. We will then incorporate those electrodes into a cartridge, which will then be used along with the reader, which we should have done by the end of January, or at least the first 2 weeks of February.
And one of the questions is a statement really that Forbes indicates that the NIH wants to hit 6 million tests per day in the US, obviously. Is there a limit to scaling of this product once approved?
Well, we already had one group ask us to produce 2 million tests. So we're working on that. But as far as scaling goes, I believe within 6 months, we could scale up into the hundreds of millions. The cartridge itself, the test cartridge, is really manufactured very quickly. And one of the things we made sure about is we made sure that this could all be made by a roll-to-roll methodology. And a roll-to-roll methodology produces vast quantities of sensors very quickly. It's probably the only way to do this, to be able to produce hundreds of millions of sensors. And thus far, we're totally adapted to roll-to-roll mechanics. Tommy, you might have something to add on that.
Yeah, I would just say that these sensors, the cartridges, are really inexpensive. Uh, they are going to be able to be produced in mass quantities because of that speed by which Greg is talking about, and the cost point for these things. I think we're going to be able to put some out there. You know, I will add also to Greg's point that not only are we optimizing now these sensors, but for sensitivity, specificity, and all of that, but we're also optimizing the manufacturability of these sensors. And we're picking, we're doing experiments with different techniques, different materials to make sure that the sensors are going to be able to be manufactured quickly and in an inexpensive way.
Greg, Greg, could you talk about post-offering liquidity for investors?
I sure can. That's been a question on a lot of investors' minds. And I just want to be very clear that the company intends to list the Regulation A plus shares on one of the Regulation A plus aftermarkets when the offering has been completed. This is called an alternative trading system, or ATS. We are in the process of evaluating aftermarket options that will work best for our investors. These aftermarkets allow Reg A investors to buy and sell securities. Because these markets are relatively new, we do not know how much liquidity they will provide. In the future, however, we may elect to list the company on a major exchange like NASDAQ. There can be no assurance that such a listing will occur. We intend to continue to grow the company and do as best we can to return a favorable chunk to the shareholders. We believe the company has incredible potential in light of the fact that the government has spent directly $25 billion on testing so far and has another $25 billion ready to go. the new bill that was passed has, I think, roughly 44 billion for testing. And that's just in the United States. Keep in mind that we're going to be selling these tests worldwide.
Okay, that's good. Thank you. Thank you. There's a question about viral load. Do we test for viral load, Tommy?
Yeah, viral load, it really tells you how much of the virus was able to be captured. So with our tests, I think eventually we should be able to give you a quantifiable number. So far right now, what we're trying to do is so that we can bring a product out as quickly as possible is really to give you a yes or no answer first and be able to get these sensors to a level of detection that then we can minutely and accurately give you viral load numbers. Um, but the first product line is going to be simpler than that.
So, Greg, there's a question about the capital table for the company. Could you explain the share structure or really the breakdown of different shares in the company? Maybe you can do that. I don't know if you're up for doing that in the video.
In front of me, but that was a question about the shareholders.
Yeah, can you explain the share structure a little better with 12 million shares being sold? Will the 12 million shares being sold come from the 50 million or will they be additional? That's the direct question.
Yeah, yeah. I believe that they're additional shares.
Yeah, they are. They are. You are issuing new shares to the investors in the Reg A Plus in addition to the prior shares that already existed.
If we meet our 50 million, that will represent roughly 20% of the company. Right.
Sounds about right. I don't remember actually. I'll accept your statement on that. Okay, so we've covered a lot of the ground. Would you either of you touch on barriers to entry for competitors? It sounds like a pretty interesting technology that's been created here.
I'll give it a quick go, then turn it over to Tommy. So far, we track and look for what's coming out because anything that comes out generally goes through the FDA. And I say that for our stockholders' benefit, and that is we can sell direct to governors of state without FDA approval. That doesn't mean that we will not get FDA approval. It just means that there is an opportunity there if it happens and this moves faster than we think. So I believe that we'll be able to sell this product very quickly, and we'll get FDA approval very quickly because many of the things that the FDA is approving right now have limits of detection that are way off the chart and involve a human eye, especially interpreting the test, a colorimetric test like the paper-based test. So Rod, did I answer that question? Or
And maybe I can give you a little bit more. The way I picture these barriers is our differentiators. So you might have competitors that are building the sensors and that they might be able to do something similar on the electrochemical sensing piece of it. I doubt it, just because there are so many elements that we're so far ahead of these, and there's still pieces of this that are under secrecy that really nobody would be able to get in there and figure that piece out, even though they would have some electrochemical tests or its electrochemical sensor. But the other differentiator is, so that's Purdue's piece. The differentiator is that IdentifySensors now comes with a rod quarter that is this, electronics genius worked at many of the famous labs that we have, and has created sensors for NASA and so on and so forth. But the genius that he brings is this reader. It's the electronic component that will then integrate with that sensor and report out into the cloud, be able to interpret that data and bring it back to you to your smartphone. Now, there might be companies that might do one or the of these pieces and parts, but not the way that we're putting it all together. They would probably, their price point would probably be much higher because they don't have it all integrated into one system, into one team like we have. This marriage between IdentifySensors and Purdue is significant, guys. Like this is, I can't stress this enough. We are really bringing the best of both worlds together. And then IdentifySensors already has a history in being able to commercialize sensors, and now we're focusing on these biological sensors. So we're using that long history, that long experience in creating products and bringing them out into the market in the sensor space, while marrying that with the readers and the sensors that we're creating at Purdue. So I really think that there are lots of barriers to any potential competitor that would think they could create this at the price point with the experience and expertise that we have. I mean, we don't need to go anywhere to do a test in a biosafety level 3 laboratory, whereas any other company, unless they have one in-house, they really have to go to specialty labs, universities like Purdue that already have all these
There's
resources.
another aspect of this, and we do have some patents that have already been issued that cover parts of this. And we filed a patent recently that is now being examined on this whole entire process and project.
So there's another question about shares outstanding and AS. I'm not sure what they mean by AS. When the stock lists, what do you anticipate the fully diluted OS and AS to be? Can you please clarify your question, and then we'll come back to that if we can. We're close to wrapping the call here. Anything you want to volunteer before we wrap, because we've got 3 minutes left on the hour.
I think maybe that question has to do with the prior answer in question.
Mm-hmm.
Are the issuing the shares that are for sale, additional shares? I believe that they are. So the total shares outstanding and issued and purchased in this Reg A would represent 20% of the company, should we fulfill our total quota of $50 million.
Yeah, I encourage, before we wrap here, a couple of important notes. I encourage you to email to Greg directly. Those of you who are here have all been invited to this webinar using Greg's email, so you can reply to him directly. If you have questions about things for Manhattan Street Capital, I'm going to put, actually Akash is going to type my email address into the box there. Encourage you to ask questions that you might have post this webinar. We will be making this webinar available as a recording with an index, so you can go back and check the details out. I appreciate everyone's involvement. I appreciate you guys in the audience showing up and being a part of this. And Greg and Tommy, thank you very much for your presentations. We're going to wrap up here in a couple of minutes. So thank you.
Yeah, thank you. And I just wanted to say, you know, it's been an amazing partnership. It's been an amazing experience so far, and I welcome all of you to be part of this journey with us as we continue to develop and perfect this technology. We must end these lockdowns, and we must bring solutions to the table that are going to allow us to move forward even under these extraneous circumstances. And I'm really proud of the work that we've done for Check for COVID and the work that we're going to be continuing to do here. I'm very enthused about it, and I welcome everybody to come on this journey with us.
I want to add one thing. Greg answered the question about liquidity. There is this new type of exchange called an alternative trading system, where the company intends, as Greg stated, to list the securities offered in the Reg A Plus on one of those exchanges. They are very simple exchanges where people can buy and sell the shares. There's no naked shorting, which to me is important, so that liquidity is, you will find out what the liquidity will be, which will be a function of the popularity of the company on the day. But the lack of naked shorting is a huge advantage to all concerned, in my perspective, in my view. Okay, so thanks everyone. This has been really good and very helpful. And look for the recording link to come through your email soon. And again, don't hesitate to follow up with email questions to Greg specifically on this. And we'll wrap up now. Thanks very much, everybody. Have a great day.